Okay, thanks so much for inviting me. I'm really happy and excited to be a part of this. Um, I think we're coming at this in my lab from a slightly different end because we're interested in developing and implementing a platform that allow us to quickly make vaccines to address future pandemics and the current pandemic in a very inexpensive and we hope effective way. So what I'm going to pose the question to is, once you guys have identified a new pathogen or a pandemic, what do you want to do about it? So what I think we can do is, using our new platform, develop a rapid, inexpensive, globally appropriate vaccine using our platform, a killed whole cell bacterial vaccine using genome-reduced E. coli. We're excited about this because the vaccines can be made in existing factories around the world for less than $1 a dose using widely available feedstocks. So I think the take home from this for the rest of my colleagues here in the P2P is, if you can identify a pathogen and a vaccine antigen target, we can produce something to test, a vaccine to test in animals in about 3 weeks, and then scale up from there. So vaccines, I think, of course, they need to be safe and effective. Ideally, they're also either universal or evolution resistant. Of course, with the COVID vaccines, we're seeing more and more problem with the new variants. But for a platform, from a platform perspective, we need to have them made fast. They need to be adaptable. Again, they need to be very inexpensive. We need to be able to manufacture them at volume using readily available feedstocks produced in existing factories around the world in many countries, and easy to store and transport. So if we have a good target and we have a good platform, I think we can have a vaccine that can address future pandemics and maybe even this one. So the technology that we're working on is a killed whole cell bacterial vaccine, which is it's synthetic biology based, so we can synthesize anything we want for about 9 cents a dose and 9 cents a base. We've onboarded our expression plasmids with Twist Biosciences. They will synthesize it into our plasmids and get it back to us in 3 weeks. It employs a 100-year-old technology, killed whole cell vaccines. It's easy to grow bacteria and then inactivate them to make the vaccine. We can benchmark it against an existing cholera vaccine that's stable for 24 months at 2 to 8 weeks. And then there's some advantages about presenting the antigen on the surface of the bacteria. One thing we use is called in our platform is gram-negative autotransporter proteins. These are a family of proteins that gram-negative bacteria have, that these bacteria have evolved to place many proteins into their outer membranes. Gram-negative bacteria have an inner membrane, a periplasmic space, an outer membrane. They have to place thousands of proteins into their outer membrane to interact with the environment. This class of proteins, the gram-negative autotransporter, does that within a single polypeptide chain. There's an N-terminal domain that mediates transfer across the inner membrane. There's a C-terminal beta barrel domain that makes a little pore-like structure, like these things here, down here in red, in the outer membrane. And then there's a central passenger protein domain that transits through that pore to be exposed to the outside world. Subject to some limitations, you can replace the native passenger protein sequence with a sequence that encodes a recombinant protein, including a vaccine antigen, and get these things to express your vaccine antigen on the surface of the bacteria. And you can put up to about 200,000 recombinant proteins on the surface of every gram-negative bacteria, on the surface of every E. coli. We synthesized a expression plasmid that has an origin of replication, an antibiotic resistance gene, and an expression cassette where we have one of these gram-negative autotransporters, a site where things can get cloned in, and it's all under the control of a rhamnose-inducible promoter, so we can ramp up expression or ramp down expression as we need to. So the overall vision is that if we find a pathogen and we get a sequence, and then we identify a particular antigen target, we synthesize the DNA encoding that antigen, clone it into our plasmid, transform it into our bacteria, and we're using E. coli. Another interesting thing that we found is that if we take a lot of genes out of E. coli, the antigens on the surface are more visible to the immune system, transform it into E. coli, optimize induction expression, inactivate it, and we have something that we can test in animals or in humans. And these some of these killed whole cell vaccines work non-parenterally, orally, or intranasally. So that's a big advantage for a global pandemic response as well. So looking at the SARS-CoV-2 pandemic, we are guided by the available sequence data. And if you look, all the vaccines except for the killed whole virion vaccines that are produced in China really express the entire spike protein of the virus. And if you map the antibodies that are made by people against the spike protein, most of those antibodies recognize what we would call an immunodominant region, the receptor binding domain that is responsible for the interaction of the virion spike protein with its receptor on the target cell. Unfortunately, while those antibodies are very neutralizing, the virus can mutate those receptor binding domains, and that's what the variants are, so that you have at least now partial escape from antibodies made against the original strains. However, if you inspect those sequences, you'll find some of those sequences do not mutate at all, suggesting that they are an essential part of the molecular machine of the virus and the fusion mechanism of the virion envelope to its host cell target. And there's a particular region there called the fusion peptide that is essentially invariant across all of the sequenced SARS-CoV-2 that anybody has sequenced so far, and in fact is invariant across all of the coronaviruses. And amino acids are in capsid are invariant for every single coronavirus that has ever been sequenced. We started a collaboration with our great colleague at Virginia Tech, XJ Meng, and he studies a coronavirus called porcine epidemic diarrhea virus. Porcine PEDV is an alpha coronavirus. SARS-CoV-2 is a beta coronavirus. They're only distantly related. However, they are very highly homologous. And we produce these, we synthesize the genes encoding these things, we cloned it into our expression system, we expressed it. Here's an immunoblot showing that. And then we have an antibody to these, and we show that we can express these things on the surface of our killed whole cell bacterial vaccines. And then we vaccinated pigs and exposed the pigs to PEDV, and we found that either the PEDV or the SARS-CoV-2 vaccine protected the pigs against clinical disease compared to a non-antigen expressing control bacterial vaccine. So these are such unrelated viruses. The implication is that if you have a vaccine that cross-protects between SARS-CoV-2 and PEDV, a vaccine targeting SARS-CoV-2 should be effective against just about any variant that the virus could come up with. Given our platform, we can use many different strategies to even enhance vaccine immunogenicity. And this is our original vaccine for PEDV. And if we multimerize the fusion peptide and then put additional hydrophilic acidic amino acids in between them and acidic amino acids on the N-terminal, we can come up with a vaccine that gives you even better binding. And we're doing the immunogenicity studies on that now. So very easy to engineer, very easy to make improvements in the vaccine antigen production that we use for our vaccine. This is a photo that I downloaded off the web. It's from Incepta Vaccine Company in Bangladesh. Bangladesh has a population of maybe 160 million, the 8th most populous country in the world. On the right here is a 1,500-liter reaction vessel. This company, using this one 1,500-liter reaction vessel, makes all the killed whole cell cholera vaccine for the country of Bangladesh and all the diphtheria, pertussis, tetanus vaccine for the country of Bangladesh. The DPT UNICEF price is 25 cents a dose. So if we can produce our vaccine and then distribute it to companies like this that exist in many low middle income countries around the world, we could quickly disseminate a way to make a vaccine all over the place, and you wouldn't have to wait for things to be produced in the developed world and then shipped around. So our take-home points are, we have what we think is a new, pretty disruptive synthetic biology-based vaccine platform that can make new candidate vaccines in about 3 weeks at a cost ultimately of less than $1 a dose. We have multiple approaches to engineer and improve antigens, and these can be made in these existing factories around the world. So we think that would be an ideal solution for a globally appropriate rapid pandemic response. So if you tell us a good target, we can make it. And this is again a reprise of the overall concept. So thank you very much.